Hi, this is podcast 5. In this podcast, we're just going to go over some pretty basic chemistry stuff. For some of you, this is way too basic. Some of you, it actually might be going too fast. The goal is just to get everybody refamiliarized with some of these terms if you haven't thought about them for a while, because they're important for things that we're going to talk about later. So if we look at these particular biologically important atoms, these aren't the only ones, but these in particular are the major players in terms of the molecules we're going to talk about. You can see that they have a different valence number, and valence are the electrons that are available to form a bond. And in particular, carbon can form 4 different bonds at the same time with one atom, and that's unusual. And that allows carbon to have lots of different properties. If you look at the bond energy in a carbon-hydrogen bond versus carbon-carbon bond versus a carbon-nitrogen bond, that tracks pretty well with on the edge of ultraviolet to visible light in terms of how much energy is in that light. And that's important for example, this idea of, does sunlight destroy the coronavirus? Well, it does if sunlight is exposed to, or the coronavirus is exposed to sunlight. So for example, if you had a mask and you wash that mask and then you put it out in the sunlight to dry, these ultraviolet bonds would break the carbon-hydrogen, carbon-carbon, and carbon-nitrogen bonds in any virus particles that were on the outside of your mask. It would take, 15 minutes, half hour for that to happen. But is that going to happen on the virus replicating inside a person's body? Well, no, because the sunlight is not going to get to the inside of a person's body. Carbon can form single bonds, double bonds, triple bonds, and rings. You see all these in biological systems. So ethane has a single bond, ethylene double bond, acetylene triple bond, and then rings, and benzene. So lots of diversity in the type of bonds that carbon can form. So you can get a lot of different shapes. This hopefully is now obvious, and you can answer this on H5P. And shape is a theme that we'll talk a lot about in this class. For the systems that make cells work and put cells together, shape is paramount in that things bond to each other and things react with each other based on shape. And even two molecules with exactly the same formula can have different shape. So if you notice this molecule 4 is coming out to your right, and then this molecule 4 is coming out to your left, these have a different shape, just like your right hand and left hand have different shapes inside the body. And there are biochemical systems that may recognize this shape and not recognize this shape, even though the two molecules have the same formula. Water is another molecule that is essential to the life of cells, and the shape of water and its properties are key to lots of biological properties of water. So this letter here is a lowercase Greek letter delta, and it means partial. So in the electrons whipping around this water molecule, they're going to spend more time around this large electronegative oxygen atom than they will around these two small positive hydrogen atoms. And so this gives this pole of the molecule, this side of the molecule, a partial positive charge, and this side of the molecule, a partial negative charge. And positive and negatives can interact with each other in hydrogen bonds. And you can see the attraction of different water molecules where positive ends line up with negative ends, such as this would be in gaseous water or in liquid water. You see this sort of positive-negative interaction, solid water also. And these are considered weak bonds, or weaker than a covalent bond or an ionic bond. It depends a little bit on the actual situation of the bond, but in general. And if you add up lots of weak bonds, you can get some important properties. So these hydrogen bonds, one water molecule binding to another water molecule, are important for lots of properties of water. Like water has really high surface tension, allows something like this water strider to walk on water. If you fill up a glass to the very edge, you can actually fill it up a little over the edge, and the surface tension of the water will curve that top surface and keep the water in the glass. Water has really high heat capacity in that it's very hard to heat up a mass of water or to take heat away from a mass of water. Practical consequence of that is if you went swimming in Lake Erie at this time of year, even though the lake water is warm relatively, it's probably at least 20 degrees colder than your body, and it will suck the heat out of you, and you're not able to stay in that lake water for very long. Another example of high heat capacity of water is if you put on a big bucket of water to boil, it takes a long time of adding heat to those water molecules until it changes phase and starts to boil. So its heat capacity is an important property that is important for cells. It is very sticky. It sticks to each other. That's really important for how plant cells transport water, for example, from roots to the tip of a tree leaf. It has its highest density as a liquid, not as a solid. So in these water molecules, as they form crystal lattice in a solid, they actually move further apart from each other. So the individual water molecules are closest together as a liquid. And as the closer you are together, individual molecules are together, is what determines your density. And so liquid water is the most dense, and solid water is less dense than liquid water. That's why ice floats in liquid water. And if you think about what the consequences of that would be for anything that lives in water, such as a bass or something like that living in a pond in Northeast Ohio, if this weren't true, that as ice would form, it would fall to the bottom of the pond until the pond filled up and killed everything in the pond. But now that ice floats, it can form a layer on top of the pond with a layer of liquid water underneath it, and things can stay alive. And water is an excellent solvent because it's a polar molecule. It can dissolve other things that are polar, that have poles, that have plus and minus ends to them. Not so good at dissolving things that don't have poles, like lipids, for example. An example of how water can be an excellent solvent: if you have sodium chloride here, you take sodium ions, and the partial negative areas of water molecules orient toward the positive ion in sodium, and the partial positive pole of water molecules orient toward the large chloride ion, and sodium chloride dissolves in water as a result of that. So water is a polar molecule. There are other molecules that will, because water is polar, will form structures spontaneously in water because they are amphipathic. They have parts of their structure that are polar and parts that are nonpolar. So if you put phospholipid in a water environment, it'll spontaneously form these bilayers where the nonpolar hydrophobic tails, the parts that don't like water, are pushed together, and the polar head groups of these phospholipids are facing the water because they're polar and water is polar also. So this will spontaneously form, and this has to do with how we think the first cells formed is by this barrier being set up by amphipathic molecules such as phospholipids. A little more structure in the phospholipid. You have this phosphate group and then some modification in this phosphate group that is it's got a polar charge to it, big negative charge here, big positive charge here, and then you have a chain of, or two chains. This is a diacyl group of a acyl chain of acyl groups, one connected to another, that are covalently linked to the polar head group. So this part here of the molecule is hydrophobic. It does not like water. This part here is hydrophilic, does like water. This would form the top half of a leaflet in a membrane, and then you'd have another phospholipid in the opposite orientation, flipped upside down, that would be interacting with these. These hydrocarbon chains in the tail aren't always straight chains, and they aren't always the same. So you could have one straight chain and one with a double bond that gives a kink, and there can be a lot of diversity in the chains of a phospholipid. In a phospholipid bilayer, this is a pure bilayer. This is not a membrane, but just a pure bilayer. Okay, a membrane is a different thing, and we'll talk about that in a future podcast. It has some chemical properties of semi-permeability all by itself. Semi-permeability meaning that some things get through easily, other things not so much. So oxygen, CO2, and small nonpolar molecules can travel through the membrane pretty easily. Water and ethanol have some ability to move through the membrane. Glucose and sucrose, lesser ability, most of it is turned away. And big hydrated ions like chloride ions, potassium ions, not going to be turned away, or not going to make it through the membrane. In a real membrane, not a just a phospholipid bilayer, but in a cell membrane that is packed full of proteins, water is not going to travel across the bilayer by itself. It's going to go through a protein pore, and same for glucose and sucrose. Those will not travel through the membrane by itself. So I want to make sure that you understand the distinction that a pure phospholipid bilayer that you pour out of a jar into a beaker of water is a different thing than a biological membrane in a cell. And the difference is the proteins in that, and the proteins that are embedded in this membrane in a biological membrane, in a cell membrane, would not allow glucose and sucrose to travel freely across, or water. When we're putting together molecules into bigger molecules, those bigger molecules are called macromolecules. Macro, just bigger. And they tend to be put together by monomers going through a dehydration reaction to form a polymer. That's not true for all macromolecules, but for a lot of them it is true. So in a monomer, you'd have a hydroxyl on one end and a hydrogen on another end. If you remove this OH and this H in a dehydration reaction, it's called dehydration reaction because you're removing the water molecule, then you can form a covalent bond between monomer 1 and monomer 2. That's a dehydration reaction. You can do the opposite, where you add water to this dimer, 2 monomer units, and that would be adding back the hydroxyl to one side and the hydrogen to the other side, and that's called hydrolysis or hydrolysis, water splitting. So dehydration, you're removing water, putting monomer units together. Hydrolysis, you're adding water, taking monomer units apart. Okay, another H5P question. Okay, so moving on to talking about macromolecules. These are just big molecules that are seen in cells commonly. Carbohydrates have a general chemical formula of a carbon-to-hydrogen to oxygen ratio. I don't care that you know about that so much. This structure on how this is written is at every angle here is a carbon molecule. So carbon number 1, 2, 3, 4, 5, and 6 in this 6 carbon molecule here. 1, 2, 3, 4, 5, and 6 in this 6 carbon molecule here. The numbering is important for some of the things we're going to talk about, and that's where it comes from, is where are they in how this is depicted. And this is just one way of depicting this. You could also just write out the formula C6H12O6 for this glucose molecule. These two molecules, even though they have the same shape, or sorry, the same chemical formula, they have a different shape. Notice this hydroxy group is pointed down on the number 1 carbon in this alpha D-glucose, and in beta D-glucose it's pointed up. How these are hooked together as monomers to form a dimer in a dehydration reaction. You can have a alpha to alpha bond called an alpha glycosidic bond. You can have a beta to beta bond called a beta glycosidic bond. And then you can have an alpha to beta called an alpha glycosidic bond. These three will have a different shape, a different 3D space shape that'll be recognized by the biochemistry that is going to seek to take these apart for energy or put them together to make a structure. So these can be used in all sorts of cells, animals and plant cells, in big, long polymer chains. Plants can use them to store energy as in the form of starch. Animals can use them to store energy in the form of glycogen. The type of molecule bond is different in the two situations. It's not always a single chain. It can be branched off as a chain, as you see here. So it can also be used to, in addition to storing energy, like the starch in a potato or glycogen in a liver cell, can be used to make a cell wall, such as in plant cell walls. It can be used to make an exoskeleton, such as in the molecule chitin, which is the thing that goes crunch when you step on a cockroach, the outer coating, the outer exoskeleton of an insect. In cellulose, which is used to make cell walls of plants and the body of the plant, when you burn a log, that wood is that you're burning is almost all cellulose, carbohydrate. The bond between individual glucose subunits is a beta 1 to 4 bond, and that's different than what you would see in glycogen, for example. And it has a different shape, and even though the monomers are very similar, the polymer has a different shape because of the shape of this bond, such that if we eat wood fiber, we can't get energy from that. In general, animals can't get energy from that. Termites can do that because they harbor a microbe that lives in their gut that can break this bond and get energy, and then they can share that energy with the insect. So lipids are not polymers, and they are hydrophobic. So it's not monomer units that are hooked together into a polymer. They are instead have a different shape, and they have the general property of not liking water or being hydrophobic. There are classes of lipids that we will run into and talk about in this class. One is fatty acids. Fatty acids are broken down by the beta oxidation pathway in peroxisomes and mitochondria for energy. They can be a straight chain of acetyl groups like palmitic acid and stearic acid. These are two really common fatty acids in mammals. They can have one or more double bonds, which does put a kink in the chain, such as in oleic acid. This is a common plant fatty acid, or linoleic acid, which has two double bonds in it. If you add together fatty acids onto a glycerol molecule, you get something called triacylglycerol. Here is the acyl chain here. There's three of them. TAG, triacyl, 3 chains. Okay, they don't have to be all the same type of fatty acid. In this example, they're not. And this triacylglycerol is the primary form that animals store fat. So if someone has liposuction to remove fat that they find that they don't like cosmetically, what is being removed is triacylglycerol. Interestingly, if we just take off one of these acyl chains, it becomes diacylglycerol, 2 acyl chains on a glycerol backbone. Okay, that's diacylglycerol instead of triacylglycerol. The Food and Drug Administration does not consider that fat. Fat is defined as triacylglycerol. And so in something like SnackWells or so-called fat-free cookies, you can use diacylglycerol to get some same sort of taste effects and texture effects that fat would give you. And you can technically call it fat-free because the FDA does not call that fat. But this is the molecule that's going to get lopped off and converted into energy by your system. And this is the one that you're going to store when you have excess energy. And so converting diacylglycerol into lots of energy, or converting diacylglycerol, just adding on another chain to make triacylglycerol, that's easily done by your cells. And so to think that they are truly fat-free is kind of silly. Phospholipids are also not considered fat. So that's another thing that is used in those so-called fat-free cookies. Okay, here's a phospholipid. We already saw this in a different shape. Here's the phosphate head group here, and then same sort of glycerol molecule, and then 2 fatty acid chains sticking off of it. So this part here, where they're just saying R as lots of different groups could be that group, and then P for the phosphate group. This part would be the charged part, and then this part down here would be the uncharged or hydrophobic part, hydrophilic part up here, hydrophobic part down there, and together this is called an amphipathic molecule, and these make up membranes. There's also steroids, which have a totally different shape, a ring sort of structure. These are also important in forming membranes. We're going to talk, when we talk about cell membranes, we'll talk about how cholesterol is needed to maintain and adjust membrane fluidity. So your membranes have to stay fluid in order to maintain membrane function, and you can adjust how fluid they are by adding or subtracting cholesterol. So cholesterol has gotten a bad rap in that in our diets, there's a common problem of too much cholesterol, but you have to have cholesterol in order to make cells. You are made of cells, and so you need cholesterol. But too much cholesterol can glom together because hydrophobic things like other hydrophobic things, and they can clog up your arteries. These acyl chains, which we have seen drawn different ways, here it's drawn just like this, and then here it's drawn with the hydrogen groups sticking out from them, and then here it's drawn in 3D. So every carbon has to have 4 bonds. So if we take this carbon, 2 are to hydrogens on either side of it, 1 is to a carbon on top of it, 1 is to a carbon underneath it. There's the 4 carbons. And if you repeat that structure, you end up with a pretty straight chain like this. But in the middle of your chain, if you have a double bond between 2 carbons, now the carbons that fall off of that are coming off at an angle. It puts a kink in the chain, and this affects membrane fluidity. So if you're trying to stack a bunch of these molecules together, it's easier to stack these together because they're all in a straight line. You put a whole bunch of straight lines together, and that makes a pretty stiff membrane. But if you're trying to stack this thing with this thing is going to require more space. That increases the spacing of the phospholipids in the membrane, makes a more fluid membrane. Plants tend to have more of these sorts of structures. Animals tend to have more of these sorts of structures. And the difference between, say, a stick of butter at room temperature and olive oil at room temperature is basically 1 double bond. So how the lipid reacts to temperature depends on how much energy is in the system and how much space there is in the membrane. And you want more space, more double bonds, less space, fewer double bonds. Neither one, you don't want to go to extreme. So if your membranes are too stiff, you can't ever open up your membrane to let in a large molecule or to excrete a protein, for example. And so that's no good. And if your membranes are too loose, then your cell falls apart. So have your membrane fluidity being just right is absolutely essential for cell function. And this is one way you can adjust your membrane fluidity. So we talked about cholesterol. We'll talk about that again. And the degree of unsaturation is another way. So this term saturated versus unsaturated, if you look at this molecule here, the maximum number of hydrogens it has. So each of these carbons can have 2 hydrogens, and it does. If you look at this one, this carbon has just 1 hydrogen off of it, and this just has 1 hydrogen off of it. So not saturated with the total number of hydrogens it could have. It's unsaturated in terms of total hydrogens it could have. So if you look at a tub of margarine, it'll say partially hydrogenated vegetable oil. And you might think, well, that's just a bunch of chemical talk. But if you break it down, it's not. So partially hydrogenated vegetable oil. They take vegetable oil, which is this, and has double bonds on it, and they add hydrogens back to it. So they partially hydrogenate it, and they turn it into this. So they take vegetable oil, turn it back into this, and as a result, it's no better for you than if you just ate animal fat. Okay, that's the end of that podcast.